All right, micro students. So lecture 2, as I mentioned from last time, is going to introduce you to the different groups of microorganisms. And so the goal by the end of this is to be able to list the 6 different categories of microorganisms that we're going to study in detail throughout the rest of the semester, and then also understand what are the characteristics of each. And so that is what we are indeed going to do. So this is the list of 6 groups of microorganisms that we want to understand. Many of these terms hopefully mean something to you. You've seen them before, like a fungus, algae, bacteria, viruses. And so again, these are the 6. We'll move through them in this order and give you a little bit more details. But one other thing that we want to understand is that there is a group of microorganisms that live in association with the human body. It's called the human microbiome or the human microbiota. That term is probably a little bit more accepted, but it's also called the normal flora. So that's another term that we use. And so these are microorganisms that are living on or within the human body. And so typically where they're found is any place on the human body that has an access point. So anything that's kind of outwardly facing or has a tube or opening to the outside is going to be colonized by microorganisms and serve as part of the microbiota. And most of the microbiome that we're interested in, that we're going to talk about, are bacteria, but realize that there are also microbiota members that belong to the group of microbes called a fungus, and then also some protozoans that live in us as well. So the microbiota is going to develop and become established during the birthing process. So as the baby is passing through the birth canal, it's going to be colonized by microbes that are present there. And then also, once it's exposed to the outside environment after delivery, then obviously the human touch of other humans to that newly born infant, and then also the feeding and the breathing are going to introduce microorganisms into the body. And so common areas of the microbiota would include all aspects of our skin, but then also our respiratory tract, our digestive tract, our reproductive and urinary tract as well. The number and the composition differs across all those areas, but that is what we are defining as the microbiota. And so now we're going to move on and talk about these different categories. And so the first group is a fungus. And so this is a list of characteristics. I'll just get them all pulled up here of what makes something a fungus. So first of all, it's one of our major categories of microorganisms or microbe, which means they are going to be small, so microscopic. But they belong to the group of organisms that we classify as a eukaryote. And so that's something that's on the review. That's something you should have learned in introductory biology. And so I would encourage you to look at, there's a review sheet of topics that are going to be really important to make sure that you have a good understanding on, that you should have learned in a previous class that you probably have to refresh. So briefly, what makes something a eukaryote is it has a membrane-bound nucleus inside of its cell. It also has membrane-bound organelles, things like mitochondria and Golgi complex and endoplasmic reticulum, etc. And eukaryotic cells tend to be larger and more complex than the other type of cell that we classify as a prokaryote. And so one thing that you want to make sure you can do is compare and contrast a prokaryotic and eukaryotic cell. So the next lecture, we're going to go into more detail about a prokaryotic cell, so we will review that then. But a eukaryotic cell structure is something that I'm asking you to review on your own. So I have notes and videos on that as well. But fungi are going to obtain food from other organisms. And so what that means when you see a mushroom growing out, there is a fungus that's growing in the soil that's extracting nutrients from usually dead and decaying matter. And so fungi do not obtain energy and nutrients from the sun like some organisms do, but instead they have to require on obtaining energy and nutrients from things that are living or have been living previously but now are dead and decomposing. And so that's what most fungi are. We call those saprophytic organisms. That's a fancy word for decomposer. But realize, as we see down at the bottom here, there's a group of fungal organisms that are parasitic, and that means that they are living on or in another living organism, and so as they're extracting nutrients and living on that organism, it's detrimental, and we can have disease that occurs. One really important thing to realize about a fungus is that all fungal organisms are going to have a structure that we call a cell wall. So this cell wall is not a characteristic of all eukaryotes. So some eukaryotes don't have cell walls, but all fungal eukaryotes do. And so that's a fundamental part of a fungus. And one other thing that we're going to talk about is that there's two major groups of fungal organisms, or at least structures of fungus. And so they can be classified as either molds or yeasts. And there's really kind of several things that you want to focus on. So notice multicellular versus unicellular. And so this deals with the number of cells that make up one fungal organism. So molds are going to be multiple cells, multicellular, whereas yeasts are just one single cell, and that is the entire organism. So that's one important difference in terms of contrasting them. And then another is how they reproduce. And so if you could remember one other word, spores are going to be how most mold reproduce. They generate these small microscopic structure called spores for reproduction, whereas most yeast are going to reproduce through the formation of buds. And so those are the two major characteristics. One other thing that we will see is that molds, being multicellular, many cells, under the microscope have these long filaments or long tube-like structures. They have a fancy name. They're called hyphae. And so that's a characteristic structure of molds only. Lastly here are the different types of fungal organisms that can cause different diseases. So disease like valley fever that we have in Arizona is important, and there's certain yeast infections like caused by the yeast candida. And then dermatophytoses is a disease that's caused by fungal organisms that land on can get onto the outer layers of our skin and cause irritation. So things like, it's unfortunately called ringworm. It's not a worm. It's a fungus. Or athlete's foot would be classified as a dermatophyte, causing a dermatophytoses. So we're going to cover fungal diseases later, but this is the first pass of what a fungus is. These are micrographs of those two types of fungal organisms. So on the right, this would be a single cell, and inside of it you can see internal structures that helps define its eukaryotic nature. So there's probably a nucleus in there and probably vacuole and things like that. But what you can also see in that one cell is there's a bud coming off of it. So that's the way so here's another bud coming off. So that's how most yeast reproduce. So unicellular budding cells, as opposed to mold. Here's that filamentous structure. So that long tube under the microscope, you can see those are called hyphae. And at the ends of hyphae, you'll get these individual structures that are spores. And so that's how most molds reproduce, is through the formation of spores. So multicellular, filamentous, called hyphae, that generates spores. So protozoan is the next group, and these are also eukaryotic organisms. So in this word, proto is a prefix for early, and zoa is related to zoo or animals. So these are similar to animals, but they're a little bit different. So they're like early, kind of prehistoric, if you want to call them that, but they still obviously are surviving. So these are single cell, so unicellular, one cell, and they are similar to animals, but they're a different group. So they're called protozoans. Many of them are found in water, again, microscopic, so you'd have to use a microscope to see them within pond water. Some do live inside of the intestine or the gut of other animal hosts, so there can be protozoans inside there. And many reproduce asexually, meaning the cell divides into two cells. Some can carry out sexual reproduction. One of the more important characteristics of these, because they move around within water, many of them have to have some mechanism, some structure that allows them to move. And so there are three major structures that a protozoan could have. So either pseudopodia is one structure, cilia is another, or flagella is the third. So on the next slide, we'll look at examples of each of those. But before we do that, these are some examples of organisms or diseases caused by protozoal organisms. So malaria, which is there's a huge disease burden across the planet from malaria. And then there's some diarrheal disease that's linked to a protozoan if you drink contaminated water called Giardia. And then Chagas disease can be transmitted through specific insects that we'll talk about later on. But protozoans are one group of microorganisms that can cause disease, and so we want to have a basic understanding of them. So these are the three examples of different protozoans that exhibit each of the three types of locomotion, or how to move. And so this is a single cell with these internal structures like a nucleus and some other internal organelles. So it's a eukaryotic cell, one organism, so unicellular. And these cellular extensions are that structure called pseudopodia that we are talking about. And so think of this as laying on the ground and reaching out your arms and then pulling yourself forward. These cells use pseudopodia to move. We have cells that do something similar like this in our body, like some immune cells will move through this amoeboid movement. Also a single cell. These, though, have little hairs on the outside, so that's a single cilium would be the word, but collectively all those little hairs are called cilia, and so they can beat and wave, and so in a fluid-filled environment it would allow that cell to be propelled through that water column. And then lastly, this extension is a flagellum, that's singular, so all of these together would be flagella, plural. And so this would be a single cell protozoan with a single flagellum on it. This is not a free living, so it's not swimming in water. These are actually red blood cells, each of these circles. And so this is a parasite that would be inside of another host, and it would be living inside of their bloodstream. So you can see this within the blood smear. But these are the three types of at least three types of mechanisms of locomotion of different protozoa. There are some animals that can cause disease within us, and so a large group of these, they're called helminths collectively. They're parasitic worms. So we're going to look at these more on the lab side of things pretty early in the semester, and so you're going to learn three groups of different parasites. Two of those major groups are categories of helminths. So these are animals, which means they are eukaryotic, so all animals are eukaryotic. All animals lack a cell wall. And some of these can be quite large. So this is a tapeworm that can be upwards of a meter or even longer in length. So not all stages are microscopic, but there are some parts of its life cycle that are microscopic. And so these are some examples of helminths. So they're different worms, so tapeworms and pinworms and hookworms and flukes and different things. So we're going to study these more in the lab section, as I mentioned. This we will talk about in the lab side of things. This is a zoomed in view of this little end of a tapeworm. So this is a tapeworm, but if you look at that little end and zoom in on the increase the magnification of it, it's going to look something like this. Different tapeworms have a different head region, but collectively this region is called a scolex. And so a scolex is what allows tapeworms to attach to the inside of the intestine. So if my hand represents the side of the intestinal wall, there needs to be something that allows the tapeworm to attach there. And so often it's a ring of teeth that can kind of penetrate and hook onto that side of the intestinal wall. And then these are not eyes, but rather those are suckers that are also for attachment. And so the head region of an adult tapeworm is called a scolex, and it's for attachment. Algae is another group of microorganisms. There are different groups of algae. Some of them are single cells that make up an organism. Some can be multicellular. And one thing that's important to note with algae is that they are photosynthetic. And so they obtain their nutrients from sunlight. And so because of that, they don't have to live and rely upon other organisms. So take example, that tapeworm. The reason it's living in association with us is it's obtaining nutrients from our intestine. So it's a parasite. But if you obtain energy and nutrients from sunlight, then you don't have to live on or in another organism to be successful. And so algae aren't going to be the cause of infectious disease, but they are a group of microorganisms, and so that's about all we're going to learn about them. They have different pigments to allow for that photosynthesis. So you may have heard of green algae, like this one has a green pigment that allows, but there's also red algae and brown algae, etc. And then another thing, because all algae are going to have a cell wall, the structure of their cell wall is going to be another important part and way in which they are going to be classified and grouped in terms of algae. Obviously, a really important group of microorganisms are bacteria. And so you are likely well aware of bacteria, but we want to have a better understanding of what they are. And so bacteria are going to be one of the two major groups that we classify as prokaryotes. So again, quick review of what that means. No nucleus inside of a cell, no internal membrane-bound organelles, so no ER, no mitochondria, etc. And when you compare a prokaryote to a eukaryote, prokaryotes are going to be much smaller and much more simple in their cellular structure. And so, all bacteria are going to be classified as prokaryotes. There is one other group of prokaryotes that they're different from bacteria. They're similar, but they're different. And so archaea is the other major group of prokaryotes. So there are only two groups of prokaryotes. There are bacteria as one, and then archaea is the other. So those are the two. So this is really looking at both bacteria and archaea. Both of them have cell walls, so that's a characteristic of prokaryotes. But what one thing we want to focus on is this term. So peptidoglycan is the building block of a bacterial cell wall. And so that's not present in archaea. It's not present in any other organism that can synthesize a cell wall. So when you think bacteria, you want to say, oh, those have a cell wall. So that's one important structure that they have. But not only that, you can say, oh, I know what makes up part of the bacterial cell wall. It's this building block called peptidoglycan. And that's only found in bacteria. It's unique to bacteria. So archaea have a cell wall, but it's not of peptidoglycan. And one other thing that we see for archaea is they are often found in what would be called extreme, or what we would think of as hostile environments for life. So extreme temperatures or extreme salt concentrations, or really deep in the ocean where there's all this weight of water on them that would be really hard to sustain life. That's where we can often find different archaea species. We don't know of any archaea that cause human disease, and so by far the group of microbes that we focus on are the bacteria, because we know they can cause disease. Archaic, we don't know of any that cause disease within humans or in our animals that we love, so no veterinary diseases either. So bacteria is what we focus on for sure. Just to illustrate this size difference, this outline that I'm highlighting right now is a single eukaryotic cell that comes from the inside of our mouth, so our cheek cell. So if you scrape the inside of your cheek, some of those cells will come off and you can put them on a slide. So that's the size of a single eukaryotic cell. That blue dot is the nucleus, this blue dot right there. But that right here, I'll put a line to it, that is a single bacterial cell. And so this illustrates the size difference. So each of these little blue dots is a bacterial cell. So this illustrates the prokaryotic versus eukaryotic size difference. The last group of microbes is very unique, but obviously very important in infectious disease. So viruses are classified as microorganisms. And the first thing that's listed here is really important in regards to understanding viruses. So all the previous groups of microbes, whether it was a protozoan or a bacteria or a helminth or fungus or whatever, we could classify them as either prokaryotic or eukaryotic, which describes how their cells look. But notice acellular means lacking cells. So viruses are not cells. So we can't classify them as prokaryotes or eukaryotes because they're not cells. So that's one thing. So that's what they're not. They're not cells. What they are is they're an intracellular parasite. And so intracellular means they have to go inside of a cell to be successful. So for all viruses to be successful, they have to not only enter a host, but they have to enter a specific cell within that host. And so what a virus is an infectious viral particle, also called a virion. And so what that means is an infectious viral particle is going to be a protein coat. And so this gray, this is a picture of SARS-CoV-2, which is what causes COVID-19. And so these little gray specks are proteins that form a spherical ball. Okay. And so that's what we call a protein coat. So all viruses have a protein coat. So proteins encircle what we call the viral genome. And so inside of this protein coat is where the viral genome is located. And so the genome is some nucleic acid, either DNA or RNA. It depends upon the virus. And so that's what all viruses are. So they're not cells. What they're going to be is a coat of protein, so basically a protein shell, and then inside of that is some piece of nucleic acid that is, once enters a host cell, so there's an intracellular parasite, that viral instruction manual, that viral nucleic acid can use the cell to replicate or make more viruses. And so there's a lot of diseases that are caused by viruses. So obviously COVID-19 that we are inundated with, but HIV and hepatitis, and the list goes on and on. I mentioned smallpox and Jenner in the first lecture, and so that's another viral disease. But there's different herpes viruses that can, like chickenpox, is caused by herpes virus, and mono is caused by chickenpox, or by a herpes virus, excuse me. So viruses are another really important category. So hopefully you have a basic understanding of, can you list the different groups of microorganisms? So there are 6 different groups that we worked our way through. And not only did we do that, but we gave fairly short one slide of what are the characteristics of each. And so that's the goal of this one. Introduce the 6 categories and have a basic understanding so you could summarize or describe their characteristics. So that's it for lecture 2. Next up, lecture 3. If I'm remembering correctly, this is going to be about how do we identify and classify microorganisms, more specifically bacteria. And so we're going to talk about that process in the next lecture.